Hello and welcome to this tutorial on foRFUN, a tool that aims to facilitate comparative genomics studies for plant species. This tool is included in AlgaeFUN, a platform that groups several tools for omics work in microalgae. Once the foRFUN tab is selected, we can see that the first step is to select the model. foRFUN allows the user to choose between an analysis based on varied plant organisms or a more general one that incorporates other rhodophyte, glaucophyte, straminophylls, and cryptophyte algae. In this case, we will choose viridiplant. If you want to change the model once you have chosen it, just refresh the page. We are going to show the typical foRFUN workflow based on the Arabidopsis CCA1 gene, for which we will study its evolutionary history and function. After choosing the model, we see how a series of organisms appear to select which ones to include in the analysis. The search can be performed by either ID or the protein sequence encoded by the gene. With this second option, we will assume that we do not have an ID of the protein we are studying. At the bottom of the screen, there are a series of tabs that organize the different analyses supported by the application. In Identify Sequences, we can search for protein sequence against our genome. If we paste the sequence of CCA1 and search for it in the Arabidopsis proteome, we can see how it returns an identifier. Then we can simply paste this identifier in the text box and select our organism of interest, in this case Arabidopsis thaliana, Mercantia polymorpha, Ostrococcus tari, and Klebsiella nitens. And then we are going to the Genes Tree tab and hit the Have Fun button. It is very important to always include the organism to which the query gene belongs, otherwise an error will be returned. The tool also allows you to choose between two ways of returning the evolutionary history. Gene trees display the gene trees organized by similarity between their sequence, while resolve gene trees displays them already reconciled with the species tree. Once the species and tree type has been selected, the Have Fun button will start the execution. We can see how the application returns a binary tree, in which the genes of each species are represented with a different color and the section is highlighted in red. A text box with the name of the genes is also displayed. Both the tree and the list of genes can be downloaded in different formats using the buttons. This tree corresponds to the gene orthogroup, meaning the set of genes in the selected model species that are descended from a common gene. The evolution of this orthogroup can be studied in the Orthogroup Expansion Contraction tab, once the previous analysis has been done. Just hit the Show Orthogroup Evolution button, and the result is a plot showing the evolution in the number of genes in the orthogroup inferred for the common ancestors of each clade, marking in red significant gains and in blue significant losses. Once the evolutionary history of the gene of interest has been elucidated, we will enter a different model, whose objective is to analyze the similarity of sequence and domains to see if a candidate gene can have the same function as another known gene. For this, PHylogenomic domains tab allows a calculation of all the PHylogenomic domains that appear in our protein. Just select the genes we are interested in from the orthogroup and start the analysis. We can see that we obtain a table with the location and description of the identified domains, as well as a representation of them on the protein chains. Now, the MCA tab allows multiple sequence alignments to be performed as a subset of the genes in the tree to identify conserved and variable parts during the evolution of the species. The result can be downloaded in either a standard FASTA format or in a more aesthetic way by coloring the different amino acids according to their chemical nature. For this, please press the Download Colored MCA. Once the previous analyses have been used to hypothesize whether two genes may have a similar function, the user tabs allow for a functional study to be performed on the orthogroup sequences. As before, the user can select as many genes as desired from the tree and choose a mode of yield terms, either biological process, molecular function, or cellular components. The analysis returns a list of the terms associated with each gene in the existing annotations, as well as two plots. The first is an association graph, which is used to see if different terms are associated with the same gene, since each node is a term and two terms are joined by edges if there is a gene in the orthogroup that has both the terms associated. On the other hand, the tree plot presents a hierarchical organization of the terms based on clustering in higher groups to summarize the result. Another type of functional annotation is provided by Keck orthology. Again, by selecting the genes to be studied, and the tool will return two main results. On the one hand, a table with information on KO terms, which identifies the genes and counts how many times that same KO has been detected in the orthogroup. Also included is information on the metabolic or signaling pathways in which the genes are involved. In this case, we see that CCA1 appears and its involvement in the circadian rhythm pathway. If Keck pathways result appear, it can be represented by mapping the gene of interest onto them. We see how our genes of interest are highlighted in red on the image. The last of the annotation is literature-based. This step performs a direct query to the plant connection tool, which returns biological relationships inferred from processing over 100,000 plant sciences articles. As with identified sequences, it is not necessary to generate an orthogroup tree to use this step, although it is intended to provide information to supplement geoterms and Keck paths. In this example, we will enter the search term CCA1, identified in the KO table, and the alias search mode, which gives you the information of the search term independently of the names used to refer to it. For more information on the search modes or to refine and extend the result of this step, we strongly recommend to visit the Plant Connection website, with more ways to represent and display the results. Once the analysis has been performed, the result is presented as a table with two biological terms connected through a type of relationship. In this case, it is referred to as interaction type. To expand or check a specific piece of information, a direct link to the paper from which the information was extracted is also provided. For example, we see how PRR7 affects CCA1 and its corresponding paper. Finally, all the proteins used in the construction of the tool are available for download in FASTA format in the Download Genomes tab. We hope this tutorial has been helpful. For any questions or suggestions, please contact us via GitHub or email.